Coming to the next presentation from Christine Moody about the benefits and challenges of using environmental nucleotides for the fish passes species detection and enumeration.
Hi, thank you all for being here. No, just okay. Today I'm going to share with you about some of the work we've been doing exploring how we can use environmental DNA and environmental RNA to understand which species move through fish passages and how many individuals are moving through. So as we're all keenly aware, you can turn the volume down almost. You put a dam in a river and it creates a barrier to fish movement. And this is a problem for migratory species that need to move from downstream habitats to upstream habitats to complete their life cycles. To mitigate this impact, fish passages are installed at non-powered and powered dams to allow fish to make their migratory life cycles. This is not only important for fish health and population and species persistence, but it's also important for ecosystem function and ecosystem services such as healthy recreational fisheries. The problem with fish passages, though, is that we don't necessarily know what species are utilizing them. Fish passages are designed for either a single species or multi-species, but we need a way to understand what is really effectively using these infrastructures. And so there are conventional methods to do this that revolve around biological assessments of downstream and upstream habitats that involve hands-on collection of fish through electrofishing or netting methods. There's also tagging methods where you can track where fish are moving to. There are also fish windows that can be installed inside ladders that allow for visual observation, species detection, and quantification. The problem with these first methods here is that they are physical hands-on. And here with fish windows, the problem is not every ladder has a window on it. And also, if it does have a window, it's not always operational. In addition to these things I mentioned, all these methods are time-consuming. They're expensive. They can be hazardous to personnel as well as the organisms that you are investigating. And so what if there was a different method that allowed you to do a non-invasive survey without having to touch the organisms, also reducing your time and reducing your cost and increasing your accuracy? This is where environmental DNA and environmental RNA can provide us with a new tool to add to our toolkit to study fish passages. And so environmental DNA and RNA is organismal genetic material that's shed all the time. So right now we're sitting in this room, you guys are all shedding, and myself, are shedding DNA and RNA. And I could collect it out of the air and sequence everybody. We can also collect it from the soil, we can collect it from honey, and we can collect it from water. The resolution that you get from DNA and RNA are potentially different, though. DNA is really stable. It's a helix double-stranded structure that can persist in the environment for long periods of time, whereas environmental RNA is only comes from living organisms, and so it gives you a snapshot in real time. And it can degrade rather quickly because of its molecular structure. And so with these using eDNA and eRNA, we might be able to capture different estimates of which species and how many are moving through, depending on how these molecular structures are interacting with the environment and what we can recover from them. And so from here, we can get at species identification, the number of individuals, as well as distributions. And so to test this, looking at the differences between eDNA and eRNA and real passages, we conducted a study in Michigan on 2 rivers, the Grand and St. Joseph rivers, across 6 dams. Some are powered, some are non-powered. And we're going to focus today on just this one, Berrien Springs, which is a powered dam, and it has a fish ladder and a fish viewing window. So we can directly compare our molecular results to visual count data. And we took samples from 3 sites, upstream site, in the passage, and downstream of the passage. And to do this, we use this backpacking eDNA sampler that allows us to filter 5 liters of water through 5-micron filters that are then put into an ambient stable solution that preserves DNA and RNA. And we took 3 samples at each site, as well as a negative control and triplicates. And then we sampled 3 events across August, September, and October of last year. With DNA and RNA, there are 2 primary methods for analysis. That is quantitative PCR and metabar coding. Quantitative PCR lets you look at a single gene for a single species, and you can get relative abundance estimates from it by looking at the relationship between copy number and an actual count number from a conventional survey. Whereas metabar coding allows you to look at multiple species at a single time, so the whole fish community, but you get read counts. And so quantifying the number of individuals from read counts is a little more precarious and needs some refinement. And so today we're going to just focus on qPCR on one single species, steelhead for cytochrome B. So here's a video from our fish window. This is a clean video and a not very smart fish. And so it allows you to see the fish really easily. This is a steelhead, so it's 1 steelhead. That's our count. Here's a dirty window. So algae builds up on these windows. The fishery managers have to go out and physically get into the ladder and clean these windows. And believe it or not, there are 2 fish here. Can you tell me which fish species they are? I can't tell you. So this creates a problem when we're using fish window to get our data. We analyzed videos all the way from August 1st through September 31st and counted the number of species that were in the videos. And what you can see, it's relatively normalized in that it's about 3 or 4 species that pass through the ladder. But the composition of the species changes. So in early August, we don't have, I keep pointing down there. In early August, we don't have a lot of species or individuals coming through. But then in mid-September, we get this run from coho salmon. The run ends rather rapidly, and we get a turnover and have a steelhead run that occurs. And the question now is, does our DNA and our RNA reflect these patterns of the run? And remember, we're just looking at steelhead. Here, what I'm showing you on the x-axis is our sampling time, August, September, October. On the y-axis is our copy number from our qPCR, with downstream being this purpley red color, in the ladder being this green color, and upstream this yellow color. First thing to notice is in October, we have the most copy numbers for both DNA and RNA for steelhead, which is great. That matches that pattern of when the run begins. And then also with eRNA, we're getting high copy numbers for downstream where we aren't seeing that with DNA. The most striking thing here, though, is that with DNA, we have 42 detections, and with RNA, we have 33 detections. There is some possibilities for this difference that will make sense. One, could it be relic DNA? Because DNA is shed, DNA lasts in the water longer. It also can come from dead organisms. And so are we picking up a signal of fish that may have moved through yesterday or a week ago, or fish that are actually upstream and the DNA particles are coming downstream? That's a possibility. The other possibility is the RNA degrading too rapidly, right? Is the RNA not reflective of our counts? Well, here is the answer. When we look at DNA and RNA with copy number, this is in a log format on the Y, and the number, our count number from our videos, with DNA, it's not working, DNA on the left here, we see that we do not have a good relationship, right? There's not a strong correlation between these two. But now when we look at the RNA, our R squared is much better. It's a significant relationship. And so this suggests that our DNA is actually reflecting that relic DNA. It's not just targeting our present DNA at that point in time, whereas RNA is really giving us that fine scale temporal view that we really need in order to count the number of individuals that are going through the passage. And so to make this more robust, we are adding additional samples at those sites. We have all those other locations. We have other target species that we're looking at, additional genes. We also did controlled mesocosms experiments with the same target species. So we know the number of individuals, we know the species composition, and we're gonna calculate the decay curves of our eDNA, and our RNA to put into our models to understand how the breakdown of these molecular components are working. And then on top of that, with all these samples, we're also gonna do metabarcoding to look at the whole fish community composition, as well as triangulating our copy numbers, our actual visual count numbers, and our metabarcoding read counts to be able to hopefully be able to model how we can use this type of metabarcoding data to estimate relative abundance of fish in the passageways. And so if you'd like to hear more about this work, please come on over to the US and join us in Michigan in August at the American Fisheries Society conference, where I will give you a full update on this. And with that, I'd like to say thank you.